I'm going to talk about a paper that I'm showing here that we recently published, where we described and demonstrate different steps of a dispersal event that took place by painted lady butterflies across the ocean. And this was kind of an exceptional event, and we all feel kind of attracted by exceptions or special phenomena in the natural world. Still, because I think it is important to know the pattern, the rule, the common, and something that it fascinates me about this species I'm going to talk about, Vanessa cardui, the painted lady butterfly. It is that it's a very common butterfly that can do very, very special things. So I promise that I will talk about this paper in detail, but first of all, I will drive you through some of the work that over the last years we have been doing, and that has allowed us to gain a deeper understanding on the migratory patterns of this butterfly. And let me also discuss about what we know about butterfly migration. What is the state of the field? And we have known for a while that there are butterflies and other insects that migrate. So the migratory phenomenon in insects is not new, and this precious book, early book by C.B. Williams in the early 30s, remains still an essential reference. But despite everything we have learned with model systems such as the monarch butterfly, the field of insect migration actually remains highly unknown and even speculative at some point at different scales. Like we know very little at the organismic level, like biological traits, also at the ecological level, movements, or at the physiological level or a genetic level. So our knowledge on insect migrations is still very limited, especially in comparison to that of vertebrates. And you guys mostly study birds, are interested in birds. So just was telling this in a note of aware that what we know in butterflies is a lot less than what you might know in birds. So there are plenty of insects that we have evidence that migrate. Among them, we know about odinates, orthopterans, lepidopterans, dipterans, even aphids, coleopterans, and even some hymenopterans maybe. But we also know that what we observe, we know that there are lots of different kinds of behaviors, kinds of movement patterns, also life histories, in a way that we cannot easily generalize what migration is in an insect. However, some common elements that most people would agree in order to define migration is that migration can be defined as a behavior at the individual scale that is typically defined like a persistent, straight, and distracted movement that involve distinct behaviors at origin and destination and a set of physiological adaptations that allow relocation energy. This definition at the individual level is conceptually great. However, we need to take into account that it's very difficult to actually quantify this in nature for insects, and it would probably require systematic experiments across many species in order to quantify these traits. In addition, this actually becomes very difficult to distinguish from other types of movements that most insects do, like it could be dispersal or nomadism, for example. So in practice, when biologists refer to migration, we typically use a set of population-level outcomes from insect movement to define these migratory phenotypes. Basically, we observe differences in the geographic and demographic scales, but not on the individual level. For example, what could distinguish migration from other movements is that migratory insects, the whole population tends to move, where other types of movements, only part of the population might move. Or that migratory movements are seasonally recurrent and therefore predictable to some extent, whereas other movements are more erratic or unpredictable. So there's probably not a systematic way to strictly differentiate migratory behavior from other sorts of movements, as there is a wide spectrum of behaviors associated to movements, and that is important limitation in insect migration. And this definition here is probably representing quite well what is the state of the art in defining migration. Migration is, like fine art, something that everybody knows about a subject on which everyone has a strong opinion, yet nobody can precisely define. I like this definition, but at the end, when we see something like this, everybody tends to agree that this is a migratory movement in butterflies, as you can see in this video, where a flock of butterflies are consistently moving in one direction, following the coastline in this case. But not all butterflies are comparable in terms of migration, and there is a diversity of migratory strategies, behaviors, outcomes in terms of movement. While the charismatic monarch butterfly, for example, sustains almost all their populations in a single majority overwintering spot, the painted lady do not hibernate, but migrates continuously between multiple breeding spots. Or an intermediate strategy could be that of Vanessa atalanta, the red admiral, which hibernates, but with some populations, different populations in multiple points. There is a huge gap also between our knowledge on insects with the state of the knowledge of migratory birds. And despite we often tend to compare behaviors in both systems, movements and biological traits in both taxonomic groups, and even we use the same terminology to define and describe, these are hardly comparable. Probably one of the most important traits that might define migration in animals is that they generally involve round-trip journeys, which also distinguish the idea of migration to that of dispersal. And the case of the insects, one important addition is that round-trip journeys involve multiple generations, meaning that this is not the same individual, but the offspring who completes the circuit. And this multi-generational scenario in insects actually adds complexity in the population dynamics of migratory insects, as the different generations are constantly shifting their range in space, typically following optimal sets of resources. But it's important to take into account that there is generally some degree of overlap in time and space in the distribution of these different generations, and therefore the generations cannot be discrete units, as it is more likely to happen in vertebrates, where seasonal ranges are more clearly delimited. Another relevant difference with vertebrates is that demographic fluctuations can be huge between generations or migratory insects. It is typical to observe in migratory species pronounced regional fluctuations, demographic fluctuations. That is, some years we observe many butterflies locally and others very few. And in the migratory context, it is hard to predict what are the trade-offs between costs and benefits. On one side, migration is typically associated with risks and uncertainty, but on the other side, the migratory strategy has obvious advantages. Overall, it is believed that about 3% of insects migrate to some degree. There are many species that migrate, but a very low percentage. So an interesting question here is, why do not more butterflies or insects migrate? And perhaps this is because these tend to be generalist species, which also involves evolutionary costly adaptations. These fundamental differences between migration in insects and vertebrates might have consequences in demography and genetic diversity patterns. These are two conceptual models representing alternative population dynamics in migratory animals. On the left, what we call a linear model, and what would be largely attributable to migratory vertebrates, with plenty of variation for sure, where populations are strongly phylopatric, leading to little mixing between them. In this model, population decreases or extinction events can lead to bottlenecks and consequent loss of overall genetic diversity. On the right, as a contrast, there is what we call a compensatory model, generally distinctive to insects, where local or regional bottlenecks are kind of diluted and compensated by success in other parts of these huge migratory ranges and population numbers. These two models represent extreme alternatives in population dynamics, leading to different demographic histories in migratory animals. And both things are migration, but both things aren't the same in terms of population dynamics. So we were wondering whether the migratory strategy in insects is successful in maintaining optimal demographic levels and genetic diversity. And we have been investigating more about how population dynamics might affect genetic diversity in migratory species. And in this comparative study led by Aurora Garcia Berro, she inferred genome heterozygosity levels along a wide range of migratory and non-migratory species. And what we learned was that actually migratory Lepidoptera tend to have higher genomic diversity than non-migratory species. And we often high levels of genetic diversity. We also observed that there's high effective population sizes and stable demographic history curves for population history. This is a pattern that aligns well with this compensatory model I was talking about, and it has obvious implications on how we look at migratory insects in terms of conservation, where there are many generalist, abundant, and often pest species which might differ from migratory phenomena that we see in invertebrates. And then where certain migratory population often appear to have conservation concerns in vertebrates. It might not be like this in insects. Said that, let's talk about this wonderful species here, which is the painted lady butterfly. Vanessa cardui is an obligate migratory species that do not have the capacity to hibernate or diapause, so they breed continuously along the year, meaning that they need to be continuously in the move. They might do up to 8 to 10 generations per year. It's a highly polyphagous species, and it is virtually cosmopolitan. Vanessa cardui is actually likely the most widespread of all butterfly species. It is virtually in all continents, except in South America and Australia. And as good migrants, they are just present for a certain period of time, where they undertake seasonal movements that are more or less consistent across years, generally doing these progressive north and south latitudinal shifts along the year to complete an annual cycle. These are even sometimes seen in very exceptional places for a butterfly, and these are the northernmost and the southernmost points where the species have ever been registered, near the Arctic and near the Antarctic. Over the last 10 years, in our group, we have been intensely working to gather comprehensive natural history data worldwide about these species, as well to gain insights on their spatiotemporal distributions. And this work has allowed us to begin an unprecedented set of studies in movement ecology, population genetics, and evolutionary genomics. And one of the very first questions we had was to resolve an important gap of knowledge regarding the Palearctic African migratory range of this species. Vanessa cardui was believed that the migratory range mainly involved the temperate zone, and that the overwintering grounds were in North Africa and Southern Mediterranean. However, it was intriguing that populations literally disappear during the winter and are very low number in Northern Africa, which doesn't make sense from an evolutionary point of view. Why the species should suffer strong population bottlenecks every winter after having prolific summers in Europe. So we tried to study this question. We came out with the possibility of a new hypothesis, which is what is the migrations after vanishing from Europe during the fall actually cross the Sahara Desert and establish in tropical Africa for half part of the year. What if they do just like the birds do? This question wasn't usual at that moment because no other butterflies are known to do this kind of trans-Saharan movements, or to live in two different places as different as the temperate region or the tropical region. So we did hypothesize it a spatiotemporal model of presence-absence and started a long research adventure over the year to test this model. We started a set of long expeditions in sub-Saharan Africa to test the hypothesis whether the species could massively cross the Sahara Desert in the autumn and actually continue their cycle all the way until the next spring in the tropical region. And we found extensive evidence of massive directional autumn migratory movement across the Sahara and breeding in sub-Saharan countries. Still, this observational evidence was not enough to prove trans-Saharan migrations. This is how then we started exploring a variety of approaches that could provide evidence of provenance in terms of migration of these trans-Saharan movements, together with our colleagues at the University of Ottawa, Megan Rich and Clement Bataille, who are experts on isotope ecology. So some of you are already familiar with the rationale behind the approaches of isotope ecology, but basically we can use isotope information from the butterfly tissues because these isotope marks are incorporated in the caterpillars through the food chain, meaning that from the soil to the plant, from the plant to the caterpillar, to the pupae, and finally to the adult, there is some imprinting information about the original isotope forms. Later, we compare this tissue data with a map or isoscape of a given isotope to infer natal origins through space or geographical assignment. While the most typically used isotope has been so far hydrogen and provides useful information mainly across latitudes, the inference still remains pretty broad. So we started exploring the use of another isotope, which is strontium, with the goal to make these geographical assignments narrower. And strontium provides information on the bedrock, thus it is highly complementary with the signal that might offer hydrogen. And indeed, when using both isotopes combined, we start substantially narrowing the geographical predictions of these migrants, as you can see in these maps. For fine isotope assignments, we also really rely on having well-calibrated isoscapes, which might be a limitation in some regions where data is limited. And this is why in parallel in these studies, we have been working on producing new isoscope models for hydrogen and strontium, especially for Africa, by including geographical data sampling for plants and also no origin butterflies that we know they are sedentary across the region. And with all these tools at hand, we have been able to unravel a lot more information about this trans-Saharan migratory patterns that happen in both directions in spring and autumn. And as you can see, some inferences in these maps here, which are pretty narrow. For example, you can see in this map here in Senegal, in these points in green. I'm not sure you can see the pointer, but where the signal clearly comes from Central Northern Europe. So that is a clear evidence of a trans-Saharan migration. So this is a representation of the conclusion of these papers, where we mostly see that autumn migrations on your right tend to be longer than and by single individuals, which might actually do more than 4,000 kilometers travel as an individual. However, movements in spring tend to be a little bit more stepwise, step by step, so by shorter distances, but still they have important barriers to cross, like the Sahara or the Mediterranean Sea. Another interesting pattern that we observe in these studies is that we realize that early migrants during the fall, they perform longer flights than later migrants. And furthermore, these longer flights correspond to butterflies that has isotopic assignments farther north in Europe. So this reminds a little bit to what is a leapfrog migration pattern that it's as described in birds, where early and northernmost migrants fly longer than later and southernmost migrants. So we have seen this interesting pattern that we believe that it's pretty consistent over the years. Especially because those butterflies that migrate the Sahara that cross the Sahara probably come from farther North Europe because the triggering factors are stronger and earlier in those part of the region of the range than not in the Mediterranean area. And from the Mediterranean area, they have more time to react to triggering factors. So they and they can also reach suitable areas in Northern Africa. So another way that we have been that we found very useful to study these migrations is actually by, sorry, let's see if you can see this video. Yeah, has been useful to us to study potential movements of these migratory species is by modeling their ecological niche. We have developed a specific approach to model migratory insects that allow us to identify regions with potential habitat where the species could breed every month. And these models are useful especially to make predictions when we don't know parts of the migratory cycle of a species. And for Vanessa cardi, for example, we still didn't know how well the distribution was in the Afrotropics. And this model points us to the potential monthly distributions of the species. So the models are useful to make predictions, and this dynamic modeling actually can extend towards a better understanding of these shifting distributions, triggering factors as well to migration and biotic interactions. And for example, test whether all generations across the cycle behave the same way in terms of interpretation of environmental factors. In this work that we are doing, we are incorporating time series satellite imagery and vegetation growth indices to improve these models. And we have learned, for example, that the green wave surfing hypothesis, for which migratory animals tend to track early resources in time space, might apply in some regions and time along the annual cycle, but not in others, highlighting other complex interactions of the environmental variables that might trigger obligate shift in distributions. But models are just models, and it is important to test their fit. So to test if the model predictions that we made are correct, we moved again into a series of field expeditions in the Afrotropics to investigate breeding presence, but this time during the winter months. And after trips in 6 countries, we explored about 240 localities, and we observed that the model is quite right. So it pointed us correctly towards the most suitable habitat for the species. And we documented for the first time breeding data for Vanessa cardi in Africa during the winter months. So we could say that we know now all the pieces of the puzzle for the Palearctic African migratory cycle. This work also allows us to identify the larval host plants most commonly used in Africa and its phenology, which is critical to understand the spatiotemporal range of the species that the species can occupy. If the phenology of the plant doesn't match, there is no way that the species can breed, which turns out not to be that diverse. Actually, this species, this diversity of plants, we tend to say that Vanessa cardi is polyphagous, and it is, but in every region they still have constraint to find their host plants. For example, in Western Africa, where we mostly record two main host plants. We also initiated a monitoring system to better understand the seasonal dynamics of the species. And this network is working, especially in Africa, where many people has been involved and working very hard to obtain this data. So far, we established around 14 permanent monitoring stations where local collaborators are monitoring the sites every 2 weeks to document both breeding and adults. And this is very valuable and unique data, considering the historical gap of research on the system in some parts of the African continent, and reveals interesting pattern. We see, for example, a clear association of the butterflies with the rainy seasons, as well as a quite ephemeral use of semi-arid countries. So with all this new data, we actually could build a new and refined model, and as a predicted and predicted as observed, we see a southwards retreat of breeding suitable habitat from the semi-arid regions in the Sahel towards the tropics. So with this, we finally locate these breeding grounds in the Afrotropics with real data and confirm the critical importance of these grounds for the species to complete their cycle. This was published in PNAS recently, and welcome to see this paper and how we unravel all this information in Africa. I know many of the people attending the seminars from there. I think you would like to read this paper. I recommend it. So we have talked about the predictability of movements, but let me talk a bit now about the predictability of population numbers. Movement and demographics are the two main factors determining population dynamics. And when understanding the cycles of migratory butterflies, it is important to differentiate between what are regular movements of the background population, the regular cycle, and what are the consequences of population outbreaks, which are exceptional. Background population follow a recurrent pattern of movement generation after generation, thus generally replicating regions where every generation takes place. Some years the numbers are higher, some years the numbers are lower, just reflecting the natural annual fluctuation of the available resources in each region. Some years, however, there might be very important anomalous events in particular areas within those large breeding regions that might lead to population outbreaks. And the study of vegetation growth anomalies might be very good predictors of population outbreaks for butterflies. And this is what this time series plots represent, where we analyze monthly vegetation growth anomalies in Africa and in the Middle East. And we're able to associate that actually right before every important outbreak happening in Europe, there was an important vegetation growth anomaly event in a suitable region in Africa that potentially could explain the extraordinary numbers of butterflies. So vegetation growth anomalies then might work as very good predictors for outbreaks, as far as these occur at the right time, in the right moment, when part of the population of migratory butterflies happen to be there, then they have the chance to explode. In spring of 2019, there was an exceptional outbreak of painted lady butterflies into Europe, which was extremely unusual. These were seen in very large numbers all over Europe, starting in the East and in the Middle East. This video, for example, was recorded in Israel in March 2019. This was, of course, a wonderful opportunity to study long-distance migrations of these swarms. So we engaged into a project where we were trying to combine different sources of information to study these movements. We collected samples during the migration outbreak across 10 countries. And for some of these spring migrants, we look at the pollen that the butterflies might have attached to their bodies. And we try to use this pollen as markers to trace their possible migratory paths, following the idea that if we might find pollen from plants that do not occur where the butterflies are collected, then we might be able to infer movements. So we isolated and sequenced pollen grains from the bodies of about 300 butterflies across 10 countries and 7 months in 2019. We molecularly identified these plants and inferred the species distribution models for them. And the results were very cool because when overlapping probability rasters from the plants distributions, we could actually detect signal of provenance when we look specifically at the plants that were absent from the butterfly sites. And this one example here shows how a group of 10 butterflies collected in Crete in April, we find signal of provenance coming from the Middle East, from Middle Eastern plant endemics. When running this exercise massively across countries, we obtain patterns that reflect pretty well the observational data from different countries. So, for example, we found contrasting patterns between the spatiotemporal groups collected in spring and in the summer, suggesting northwards and southwards movements, respectively. So in other words, butterflies collected in spring in Northern Europe were carrying pollen from plants distributed in the south, and butterflies collected later in the summer in the south were carrying pollen from plants distributed in the north. And putting all the evidence together, we concluded that a timely anomaly of vegetation growth that happened in the Middle East produced a memorable outbreak of butterflies that massively invaded all Europe northwest, and that a reverse migration in direction occurred in Western Europe, which explained the later observation in the season, and that this outbreak was noticed even several months ago and generations later in places as far as the Canary Islands or Cameroon, according to our monitoring data by African collaborators. Note that the effect of this outbreak was noted as well in Asia, so do not view this as a northeast movement as a unique response. Overall, these results illustrate how large migratory ranges and their associated populations can be. So let's finally talk about the transoceanic case of dispersal. This is actually why you invited me for, so sorry about all the rest. I'm going to tell you that Vanessa cardui is absent in South America. It's a continent which it does not occur generally. Still, there were a very few records, fewer than 10 to my knowledge, that were cited in the northern coast in French Guiana or in the Guiana. And based on those records, I decided to travel to French Guiana with the hope that I could find immigrants arriving to the coast. And I couldn't find any after many days of exploration, but on the very last day of my trip, while surveying one beach, I was really impressed to see how several individuals of painted lady butterflies were standing on the sand on the beach, totally exhausted and pretty destroyed by the appearance of their wings. And that was really exciting because what happens where do we think these butterflies were coming from? So the question was, or the possibilities were two. They could be from the closest populations in North America, or maybe they were African. To test this hypothesis, they could also be local, that we just missed the information that we were breeding in the region. So to test all these hypotheses, we ran through a process of various analyses that most of them I explained in previous cases, but we put them together for this specific case here. And starting from genomic data, I analyzed a bunch of individuals from populations both in the old world and in the new world. And what the genetic tells is that these individuals in South America do not cluster genetically to North American populations, but instead this cluster with the old world genetic pools. So the hypothesis of an African origin of these migrants starts gaining weight here, but still it's hard to believe, really, from Africa, what does it take for a butterfly to cross the Atlantic Ocean? So we kept exploring, and we started looking at the winds. We did model the backward wind trajectories arriving to the French Guiana coast for that particular date of the observation at different layers of the atmosphere from 500 to 2,000 meters. And what we observed is a consistent pattern of winds at different altitudes that were coming straight from West African coast, and that those currents originated 200 hours before reaching South America. Actually, these favorable wind trajectories were only consistently reaching the African coast during the 2 immediate days prior to the observation. Not the week before, not the week after, looking quite a unique scenario for wind-aided or wind-assisted transatlantic dispersal. So the African origin becomes highly possible here. But what makes an Afrotropic origin certain is that we could sequence pollen grains from 3 plants or 2 plants that are endemics from the Afrotropics. So yes, these butterflies crossed the Atlantic because they visited plants that only grow in Africa. But still, we keep exploring because we wanted to know, where did they grow up? What do the isotopes suggest? And what the isotopes suggest is that these butterflies might come as far as from Western Europe or in some region of Africa, but that considering the timing that are likely to be Trans-Saharan migrants, increasing the individual migratory distance up to 7,000 kilometers, which would represent the longest flight known for any insect. So we actually identify the most likely regions of natal origin if we combine also the information from the isotopes with the probability rasters of monthly distributions from the spatial-temporal niche model that we performed earlier. And these narrow our hypothesis to regions in Western Africa, like in Ireland, UK, France, or Portugal, and to lower extent, some regions in Mali, Senegal, or Guinea-Bissau. So to summarize, we have been able to trace back the migration history of some butterflies that were collected in South America, that crossed the Atlantic aided by winds, that made a stopover in the Sub-Sahara, but that grew up most likely somewhere in Europe. So in total, more than 7,000 kilometers journey crossing a sea and desert and an ocean. So this proved that combining multiple evidence, we can learn about long-range insect dispersal in an unprecedented way, and that we should probably invest more efforts on investigating this phenomenon given the ecological relevance that it has. We also performed some energetic flight models that considering the variables like the speed of the wind, the speed of the butterfly can take by self-assisted flight, or the fat reserves that they might have. And we concluded that this flight happen between 5 and 8 days of non-stop flight across the Atlantic. We made some calculations that if there wouldn't be wind, they could be only able to fly for approximately 800 kilometers. And even with wind assistance and only active flight, that means flapping the wings, they could only reach almost 2,000 kilometers. So that means that a minimum effort strategy is required, which means just flapping the wings what is necessary not to fall, but also just gliding and using the wings with minimum effort. And all this could also only happen if they started having at least 14% of the fat reserves, not less than that. So with this, I hope I've convinced you that long-range movements in butterflies is a very cool phenomenon to study, and that we might be actually underestimating or seeing only the tip of the iceberg of what migratory butterflies can potentially do. I hope also convince you that we need plenty of data of different sort to nicely reveal complete interconnected patterns of insect migration. And I would like you to check our citizen science initiative to move towards a global monitoring system for migratory butterflies. Please feel free to download our phone app, and you'll learn how to contribute monitoring data, especially documenting breeding sites of some different species in your region. And this platform will be also soon linked to a website where it will be easier to download data, protocols, as well as to communicate with other users. And I would like to really thank this wonderful team of people that I'm very fortunate to work with, this enthusiastic young team of researchers in my lab. And also, of course, a plenty of wonderful collaborators all over the world that are making possible these difficult studies that involve many countries and many observers, and also the funders that are making possible this research over the years. And with this, I think I'll take questions and let's discuss more about anything you would like to. Thank you for your attention.
Gerard Tanavera, thank you very much for this wonderful presentation. And I think now is the time for a very small translation into French by Matthew. I hope he is here. So yes, Matthew,
Yes.
the floor is
Hello,
yours
everyone.
for 5 minutes. Thank you.
Ok. Donc, je vais faire un résumé rapide de la présentation assez dense de Gérald Talavera. Cette présentation portait sur plusieurs travaux de recherche menés sur l'écologie de la migration d'une espèce de papillon. Et donc, le nom français de cette espèce, je pense que c'est la belle dame ou Vanessa Deschardons. Vanessa Carduy, c'est son nom scientifique. Et donc, ces recherches décrivent la migration de cette espèce sur plus de 4000 kilomètres à travers l'Atlantique. Donc, c'est une espèce qu'on retrouve à la fois en Europe, en Afrique de l'Ouest et qui a du coup été aussi identifiée en Amérique du Sud. Alors, plus généralement, il y a quand même beaucoup d'espèces d'insectes qui sont des migrateurs. On estime qu'il y a environ 3% des espèces d'insectes qui migrent dans le monde. Et une des particularités de la migration chez les insectes, contrairement à la migration chez les oiseaux, c'est que chez les insectes, cette migration peut se faire sur plusieurs générations. C'est-à-dire que les individus qui vont, par exemple, quitter l'Europe et atteindre l'Afrique ne seront pas forcément les mêmes qui vont plus tard revenir en Europe. Ils vont se reproduire en Afrique et c'est les générations suivantes qui vont faire le voyage inverse. Donc ça, c'est une des particularités qu'on va trouver chez les insectes, c'est cette migration transgénérationnelle. Hum, Une autre aussi des particularités, c'est que vu que la migration se fait sur plusieurs générations, les populations en fait vont varier en taille en fonction des générations, et cela va dépendre des ressources notamment qu'ils trouvent sur les sites où ces générations apparaissent. Et donc pour en revenir donc à Vanessa Carduy, c'est donc une espèce migratrice qui se reproduit toute l'année. Et donc, ils ont fait plusieurs études pour essayer de comprendre les patterns de migration de cette espèce et aussi avoir des données concernant l'évolution de la taille de ces populations. Donc, ils ont notamment utilisé les isotopes d'hydrogène dans les tissus pour déterminer, par exemple, l'origine géographique des individus qu'ils avaient capturés dans différentes parties d'Europe et d'Afrique. Et ils ont trouvé que, notamment, les populations donc d'Europe pouvaient migrer en Afrique de l'Ouest en traversant le Sahara. Ils ont aussi identifié que les populations qui y viennent qui sont d'origine du nord de l'Europe vont migrer plus tôt et vont mettre un peu plus de temps pour réaliser leur migration vers l'Afrique. Ils ont aussi modélisé les régions de présence potentielle et fait des prédictions donc sur toute l'Afrique tropicale. Puis ensuite, ils sont allés sur le terrain pour vérifier si on retrouvait bien donc dans ces zones de présence potentielle les papillons et si ce sont des sites de reproduction. Ils ont du coup en Afrique aussi essayé de chercher sur ces sites de reproduction quelles étaient les différentes espèces de plantes hôtes pour les chenilles. Et concernant la démographie et donc les variations de la taille des populations, euh, donc ils ont notamment mis en évidence que on pouvait prédire l'évolution des tailles de population en fonction de la croissance végétale. Puisque quand il y a par exemple des conditions favorables et donc que la végétation en Afrique se développe rapidement, ça va être corrélé avec une augmentation de la population de ces papillons. Et donc, il y avait par exemple un cas qui avait été étudié en 2019, où on avait constaté une explosion de la taille de population de ce papillon au Moyen-Orient. Et ensuite, on a observé quelques mois plus tard donc l'arrivée massive de ces papillons en Europe. Donc, cela montre qu'il y a une connexion entre l'évolution des populations à un endroit géographique et ça a une incidence sur les autres endroits où on va retrouver ce papillon. Euh, et donc, vers la fin de la présentation, ils ont aussi démontré donc, la présence de cette espèce de papillon Vanessa carduy en Amérique du Sud. Et donc, ils ont essayé de savoir quelle était l'origine de ces individus. Est-ce que c'était des individus qui venaient d'Europe et d'Afrique? Et donc pour cela, ils ont dans un premier temps modélisé un petit peu les vents pour savoir si c'était physiquement possible pour ces papillons de traverser l'Atlantique. Donc ils ont vu un petit peu les différents courants d'air et ils se sont rendus compte que c'était théoriquement possible. Euh, Ils ont aussi détecté, et donc c'est ça aussi qui a confirmé l'origine plutôt africaine-européenne, c'est qu'ils ont détecté la présence de pollen de plantes africaines sur ces papillons. Et donc, encore une fois, une analyse des isotopes ont révélé que les papillons qui avaient été retrouvés en Amérique du Sud venaient notamment d'Europe et d'Afrique de l'Ouest. Voilà. Et pour finir donc sur le résumé, à la fin de la présentation, il a aussi montré qu'ils sont en train de développer une application dédiée justement au suivi en temps réel de la migration de plusieurs espèces de papillons. Donc ça, c'est une application un peu comme eBird qui est destinée au grand public, qui sera téléchargeable sur smartphone et bientôt il y aura un site internet de développé où chacun d'entre nous peut, s'il arrive à reconnaître une espèce de papillon migratrice, entrer du coup le nom de l'espèce puis la présence à un endroit donné. Voilà, merci. Thank you.